All right, today we're going over Unit 8, which is our meiosis, which is also they're going to be talking about these chromosomes and the karyotypes and also some different types of mutations. So let's take a look. Um, a key thing here in this is our differences between mitosis and meiosis. We talked about mitosis already. These have 46 chromosomes inside of it, and then we divide one time. So this is a division, and we divide one time and we create two cells. Again, they're going to be identical and they're going to be the same. For meiosis, we're going to divide twice. Here's our first one, and here's our second time of dividing. And when we divide two times, that allows us to become haploid and get sex cells, which would be sperm or egg. So let's take a look at this. In two key types of definitions would be diploid and haploid. So human body cells, on your notes, you're going to put that they have our body cells, like our skin or our nails or our hair, all have 46 chromosomes inside of each cell. And what we call that is diploid. Di meaning two. That would be two chromosomes at every single number for our cells. We call these somatic cells. Our body cells are somatic cells. And then we have sex cells. And these sex cells, like sperm and gametes and eggs, all of these only have 23. We call these haploid, or what I like to call is half the total number. So how do our sex cells get from 46 to 23? Because our human body cells have 46. So how do these two, sperm and egg, get from 46 to 23? We call that meiosis, and we call that dividing two times. So let's take a look. Again, here's a sperm cell with all of our DNA inside of it. In that sperm cell, this is one individual cell. It has 23 chromosomes in it because it is a gamete, and that is also what we determine as haploid. Same thing for an egg cell. When they join together, they form a baby zygote, which is actually down here. And what we call this is having 46, because this 23 and this 23 from mom and dad have joined together, and now we have a diploid organism. So we refer to a couple things throughout this meiosis plan, and we have parent cells versus daughter cells. Parent cells is coming from the very first one. Parent cells have 46 chromosomes. And usually they're identical to all other parent cells. Daughter cells, on the other hand, have 23 chromosomes. So if we say there are 4 daughter cells, each one is going to be different from the parent cell and also different from its siblings there before. So at the very end of meiosis, we end up with 4 daughter cells, each one genetically different. So mitosis and meiosis. Again, some more differences here. These are very important, obviously. But mitosis, we refer to body cells. Meiosis, we refer to gamete or sex cells. In mitosis, we've got diploid cells, all having the total number of chromosomes inside of every single cell that a human should have. And then we call these the daughter cells. They're going to be identical. They're not going to be different. And then there's only one division. In meiosis, there are 2 divisions. So we end up with a total of 4 cells at the very end of meiosis. And then each cell is different from the parent cell and from each other. So nothing is ever going to be identical unless there's very rare cases. Not rare, I'm sorry. Very different cases of twins, and that is a different story. Lastly, we have them being haploid, so they have half the total number, and here is what that looks like. Again, mitosis going through only one division, and I'm talking about division of the nucleus. Here's our nucleus right here. It is divided, split up, and divided one time to give us 2 nuclei. And that, again, would be identical over here. Sorry about that. But on here, on this side, we've got two divisions of the nucleus. So for our first one, we have this nucleus right here going away. It's gone, but then it comes back right here. So that's the first division of the nucleus. Then again, this nucleus disappears so that we can split apart these chromosomes and this DNA, and it is put into a second division for 4 different nuclei, which we call haploid, and they are all different here. They are all different here. And so what does that look like? We have 2 different types of meiosis. We have meiosis 1, which goes through prophase, metaphase, anaphase, and telophase, all 1. Prophase, metaphase, anaphase, and telophase 1. And then we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Each one doing the same thing, but separating different things. At the very beginning, we have all of our DNA sitting inside of one. So what we have are homologous chromosomes. Homologous chromosomes sitting right next to each other, just like this. In prophase 2, these homologous chromosomes are actually finally separated and put into 2 different cells. So now we have a full X over here and a full chromosome over here. And finally, in prophase 2, the second division of our nucleus, each of these chromosomes are now separated by their chromatids. So now there's just going to be one shape in either side. So again, these chromosomes are being separated. We start with homologous chromosomes, full chromosomes sitting next to each other. After that, they're going to be separated. That is one division. Then after these are sitting in the same cell, we divide them one more time. In anaphase, the chromosomes are pulled apart, and we got a chromatid sitting in each one. This gives us only 23 pairs in every single cell. So here's what that looks like again. And one thing I want to point out here is that we have one of the most important times in meiosis is sitting right here. This is our prophase. The reason I can tell it's prophase is because there's a nuclear envelope sitting around. There are full chromosomes sitting in there as well. And these chromosomes right here are actually beginning to pair up. What we call this is synapsis. And in synapsis, homologous chromosomes pair up side by side, and the next thing that occurs is something called crossing over. And during crossing over, these chromosomes actually decide to cross different parts, and they will break off and join the opposite chromosome so that each of these cells over here at the very end can be genetically different from each other. And they cross over at random places, and this gives us variation. So if we take a look at that, there are 3 main ways to add diversity to daughter cells in meiosis. One of those is crossing over. The other one, what we call, is independent assortment, meaning during metaphase, when the chromosomes line up in the middle, independent assortment means that one independently, a chromosome can line up in the middle, and it doesn't matter where they go. They randomly assort each other, top, middle, and bottom. Lastly is random fertilization, and that just means randomly it is a choice from a sperm cell to fertilize an egg.